Okay, hi everyone. This is lecture number 4, DNA Part 1. So we are going to do DNA in 2 separate sections because there's quite a bit to go over, and I don't want to overwhelm you guys. So we're going to start with Part 1, which will be the basics of cell division, DNA, and replication of DNA. So cell division is the process of mitosis, and in sex cells, or for gametes, it's the process of meiosis. To understand mitosis and meiosis, we also have to get into a little bit about chromosomes. We'll then get into the structure of DNA, so we can talk about how DNA is copied, or DNA replication. Part 2 I will post in a separate video, and that will be protein synthesis. Okay, so all organisms grow and reproduce, and as humans, we are organisms. This happens cell by cell, tissue by tissue, passing on information to the next cell so that each cell and tissue can be copied. So this is happening as we grow, when we are younger, when we're children. This also happens as we grow new structures. So when your hair grows, when you replace cells in the body, like you replace skin cells, things like that. All of this requires reproduction and recycling of cells. So the cell cycle is one round of making new cell copies. The basic process involves copying DNA and cellular machinery, and then splitting the copies of DNA and the cellular machinery to form 2 separate cells. So let's look at how this happens. We're going to talk about 2 main types of cell division. First, mitosis. Mitosis is cell division in the majority of cells. In humans, this is all somatic or body cells. So soma refers to body, so when we say somatic, that means body cells. Then meiosis, which is cell division in reproductive cells to form gametes. In humans, those gametes are eggs and sperm. So the cell cycle has 6 stages that we're going to learn today. Interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. Technically, cell division is a part of cell reproduction. Cell division is steps 2 through 5, prophase, metaphase, anaphase, and telophase. And that is the process of mitosis, or cell division. So to understand cell division, we first have to talk about chromosomes and get some terms out of the way so we're all on the same page with our terms. So DNA is packaged in chromosomes. Hereditary information is stored in DNA and tightly packaged in chromosomes. A chromosome is a single, long, double helix of DNA, which is wound tightly together with proteins. At the center of the chromosome is a centromere that holds the chromosome together. So this is a chromosome. It is DNA wrapped in protein with a centromere at the middle. So chromosomes are DNA wrapped in proteins. RNA will be nearby to assist with reading the DNA for protein synthesis, and we'll talk about that in part 2 of the DNA lecture. One long strand of double helix DNA, remember that DNA is in a double helix, I'll show you that again in a bit, is continuous for the length of a chromosome. And on average, it's going to contain about 140 million nucleotides. Remember that nucleotides are the building blocks of nucleic acids, which includes DNA. In other words, there are thousands of genes, which are strings of nucleotides encoded on a single chromosome. The proteins on the chromosome are going to help to coil the DNA tightly so that it has a compact structure within the nucleus. So here is an example of human chromosomes that have been stained. Humans have 23 types of chromosomes. We number them 1 through 22, so that's numbered over here, 1 through 22. And the 23rd we label as X or Y. So numbers 1 through 22 are the autosomal, or the regular body chromosomes. X and Y are body chromosomes that are found in different combination in males and females. So everybody has 1 through 22. And then depending on whether you're male or female, you will have a different combination of X and Y. So if you're male, you will have XY. If you're female, you don't have the Y chromosome, you have 2 copies of the X chromosome. So who's this? Is this male or female? Yep, these chromosomes came from a male because there's an X and a Y, but all males and females have numbers 1 through 22, the autosomal chromosomes. And these chromosomes will be found in all cells of the body. Human cells actually have 2 of each type of chromosome. Did you notice that on the previous image? Go back and take a look if you didn't see it. I'll show it again in a second. Human cells, in other words, are called diploid. Di for 2, ploid for ploidy, diploid, 2 types, 2 of each type of chromosome. So we have 23 types in human cells, and then 2 of each type for 46 total chromosomes in every cell of your body. The 2 chromosomes of each type are called homologs or homologous chromosomes. So homologous chromosomes, you get 1 from your mother and 1 from your father. So here we have the maternal homolog, and here we have the paternal homolog. We put those together, that is 1 pair of a single type of chromosome. So let's look again at the 23 pairs of human chromosomes. So here I'm circling 1 of them. There's a homologous pair, so there's 1 from your mom, 1 from your dad, and you inherited those, each pair, for 23 total pairs of chromosomes. 46 chromosomes in every human cell because humans are diploid, or 2 copies. Two pairs. One pair of each type of chromosome. So humans are diploid because 1 homologous chromosome is inherited from the mother, 1 homologous chromosome is inherited from the father. Homologous chromosomes are chromosomes of the same type. In other words, they encode the same genes, or the same types of genes, but the information will be different depending on what is inherited. What does this mean? Here's an example. Chromosome 1 contains the gene for eye color. The maternal homolog will encode green eyes from the mom. The paternal homolog will encode hazel eyes from the dad. So I'm totally making this up. I'm actually not sure if eye color is on chromosome 1 or not. And eye color is actually more complicated. But as an example, chromosome will have a particular type of gene. 1 homolog will have 1 variation of that gene, or say green eyes. The other homolog will have another variation of that gene, or say hazel eyes. And then what you get is a combination of maternal genes on 1 chromosome, paternal genes on another chromosome. Each will have their own variations depending on what is inherited from mom or from dad. And then the child will end up with some combination of that. So this is the case of me and my husband. I have green eyes. My husband has hazel eyes. And then our first baby actually turns out our second baby too. They both have blue eyes. So some combination of genes that gives a different variation in the child. So now that we understand the basics of chromosomes, let's talk about what happens when chromosomes are copied or replicated. So before we can replicate cells, we have to replicate the chromosomes. So before cell division or making new cell copies, chromosomes must be replicated. A copy of each homolog is called a sister chromatid. So we start with the pairs in a normal body cell, and then we're going to copy that body cell. We then need to copy each type of chromosome. So to do that, we go through the process of DNA replication. We'll go through those steps in a bit. And we make copies of each type. So here are your maternal copies, and here are your paternal copies. So here we still have homologous chromosome pairs, but now each pair has a twin. So the twin sisters, or the copied homolog, are called sister chromatids. Let me say that again. We start with normal, regular, homologous chromosomes in a body cell, one from mom, one from dad, for the individual. We then replicate the maternal and paternal homologs to create replicated or duplicated chromosomes. Each copy is called a sister chromatid. So we will have sister chromatids from the maternal copied chromosome, sister chromatids from the paternal copied chromosome. Got it? Okay, draw it out if this is confusing for you. So we have homologous chromosomes in body cells. In order to copy those body cells, we have to copy the chromosomes. We will replicate the chromosomes, so we have sister chromatids from the maternal chromosome, sister chromatids from the paternal chromosome. Okay, now we understand enough about chromosomes that we can talk about the cell cycle. So in animals, or in humans, we're eukaryotes. Eukaryotic cell cycle has 6 stages: interphase, prophase, metaphase, anaphase, telophase, cytokinesis. Again, the division of the nucleus technically is mitosis, and that is occurring in stages 2 through 5. So let's start with stage 1, which is interphase. Interphase is the preparation phase, or the cell performing its normal daily life. So in my mind, this is like your life in your 20s, when you're out having fun, doing your normal everyday life, until you decide to have kids. So it is a long period of the cell performing its daily functions, and then setting the stage for cell division. So interphase is the longest portion of the cell cycle, and it's the daily life of the cell until the cell needs to divide and replicate. So over here, what I'm going to show you for each of these phases is a picture of the cell with the nucleus. Keep track of the nuclear membrane, which is the membrane around the nucleus. Keep track of the chromosomes, maternal and paternal homologs, and keep track of the centrioles, which are mitotic organizing centers, which will put out mitotic spindle to support mitosis when mitosis begins. Also, up here is an actual picture of the nucleus of a cell during division. So during interphase, chromosomes will do their normal daily life, and then at the very end of interphase, chromosomes will replicate. So we'll form sister chromatids for each homolog, and then chromosomes will begin to condense. And when chromosomes condense, they become slightly more visible. The sister chromatids that have now been replicated will be held together by centromeres. And the centrioles, which are the organelle for mitosis, will be replicated in preparation to make the mitotic spindle. What's the mitotic spindle? Here's the mitotic spindle. See this orange structure here? This is a microtubule structure that will support the chromosomes as they are moved around during this process of cell division. So the next phase is prophase. Prophase is technically the beginning of cell division, the beginning of mitosis. And chromosomes are going to condense so much that they are actually visible. So take a look at this picture of an actual cell. You can now see them rather than just sort of that fuzzy blue that you saw on the previous slide. You can now see individual chromosomes being pulled apart. Remember that these are duplicated or replicated chromosomes. So now you'll see there's a maternal homolog with one sister, another sister. There's a paternal homolog with one sister, another sister. So there are sister chromatids are now present because the the DNA was replicated in interphase. The nuclear envelope is going to start to degrade around the nucleus, and the chromosomes will then take up the majority of the inside of the cell here in this picture. The centrioles are going to move to opposite ends of the cell and begin to form that organelle for mitosis, the mitotic spindle. That's going to be used to grab onto the chromosomes and pull them apart. So the mitotic spindle fibers will attach to the center of the chromosome at a region called the kinetochore. Now we're going to start to actually grab onto the chromosomes and move them around. So in the next phase, stage 3, metaphase, the chromosomes will begin to align at the center of the cell. So notice there is no more nuclear envelope, and the chromosomes are aligned here at the center of the cell. So all down the middle, okay, and then here in our regular picture, all down the middle, the mitotic spindles are attaching to the kinetochores, or the center of those chromosomes, and they are now fully extended toward the opposite ends or opposite poles of the cell. So there are the mitotic spindle fibers extending towards the opposite poles of the cell and holding the chromosomes at the center of the cell. Next is anaphase. In anaphase, now we're going to divide the chromosomes and send half to one side and half to the other side. So we'll take each sister and send one sister to one side, one sister to the other side. So in anaphase, we have the separation of sister chromatids. They will be split at the kinetochore by enzymes that you don't need to know about and pulled to the end of the cell by the mitotic spindle fiber. So we had the sister chromatids lined up in metaphase, and then they separate, so you'll have one copy of mom on one side, one copy of mom or maternal homolog on the other side, one copy of paternal homolog on one side, one copy of paternal homolog on the other side, separating the chromosomes to the opposite ends of the cell. The next phase is to reform the nucleus around each set of chromosomes. So this is telophase, step 5, and this is when the nucleus is reformed around each group of chromosomes. The nucleus is formed around each side, on either side of the cell. You have identical copies now of the nucleus on either side. The mitotic spindle has done its job. It has separated the chromosomes to either side, so it will start to degrade. The nuclear envelope reforms, the nucleolus reappears, and mitosis, or the division of the nucleus, is now complete. But you can see that we still only have one cell. It's starting to pinch inward a little bit, but we only have one cell. So what needs to happen now is the last phase of the cell cycle, in which you form a completely new plasma membrane around each nucleus. So this is cytokinesis, or division of the cell membrane to form two final cells, division of the cytoplasm as well. So in animal cells, this happens by pinching the two centers, by pinching at the center of the cell and forming two totally new plasma membranes. That pinching is called a cleavage furrow, and it forms plasma membranes surrounding each replicated and divided nucleus. So now we have two identical daughter cells, which are now identical copies of the original parent cell with the same number of chromosomes, the same types of chromosomes, and the same complement of maternal and paternal homolog chromosomes in each cell. So I have summary figures for you guys to review just while you're studying here. And let's talk a little bit about cell cycle control. So most of us know someone, even some of us have experience personally with cancer. So in research, we study the cell cycle and get a lot of funding to study the cell cycle for cancer research. So cell cycle control is a very important part of this research. So the cell cycle contains checkpoints throughout the different phases to make sure that this process is going the way that it should. So these main checkpoints we call G1, G2, and M checkpoints, where we check the progress and the health of the cell at times during the cell cycle. And if anything is going wrong, a decision made by molecules in the cell is made to pause, stop, or if everything is going well, continue the cell division process. So G1 is a checkpoint during interphase. That's a checkpoint for cell growth. Is the cell growing properly? G2 is a checkpoint for DNA replication. This is to make sure that the DNA was faithfully copied in order to continue on to mitosis. The M checkpoint is during mitosis, and it is to make sure that the separation of the chromosomes is going properly. If that doesn't happen, the cell can exit mitosis and stop cell division. If the cell then completes mitosis, it can begin cytokinesis and continue. A cell paused and not growing or dividing because it is in the process of going through the checkpoints or being repaired during the checkpoints is said to be in G0. This is a diagram of the normal cell cycle where we have the G1, G2, and M checkpoints. So what is cancer, just quickly? And I don't normally test you guys clinically on these types of questions, but cancer is so common and it comes up so often that I want you to be familiar with cancer. So cancer is uncontrolled growth. In other words, unchecked mitosis that can spread invasively to other parts of the body. So a mass of uncontrolled mitotic cells is called a tumor. These are cells that are growing and dividing without being properly checked. Benign tumors are enclosed by surrounding tissue. They don't spread and they're not invasive. Malignant tumors are not encapsulated and they can spread to other parts of the body. Cells that leave a tumor and spread can form new tumors at distant sites called metastases. That can be very dangerous because then you can have many tumors wreaking havoc around the body. Chemotherapy actually targets the cell cycle, and this is where you hear about in chemotherapy some of the major side effects are because chemotherapy stops division in any cell, not just a cancerous cell. So this is why hair growth stops. You have stomach issues during chemotherapy, and because of that, the side effects of chemo can be very severe because the growth and replacement of normal cells is important throughout life, and if that is stopped by chemotherapy, then digestive system, skin, nervous system, bones, muscles, a lot of issues and side effects can happen through stopping cell division in the body through chemotherapy. Okay, let's now touch on the process of meiosis, which is cell division in reproductive cells. So reproductive cells are, their purpose is to form gametes. Gametes will have half the complement of chromosomes. In other words, they will be haploid. So that way, mom can pass on half of her genes, and dad can pass on half of his genes, and we put them together to make a new individual. So in the female, those haploid cells containing half the complement of mom's genes are called oocytes or eggs. And in males, the gametes are spermatocytes or sperm. Each gamete has a single copy of 23 chromosomes. And then when you get the egg and the sperm, or the female and male gametes, together, then we will have 46 total chromosomes in the final new individual. So during the sexual reproductive process, gametes will fuse to form a zygote. The zygote will then undergo repeated mitosis to grow into an embryo with body cells containing the full complement of 23 pairs of chromosomes, just like the mitosis we just went through. But the process of creating gametes in the mother or in the father is the process of meiosis. So here is a picture, example of these haploid gametes. So here's the haploid egg with the female homologs. Here's the haploid sperm with the male homologs. They would combine together, so you have maternal and paternal homologs to make a full new diploid individual. And in this, during development, is called a zygote as it is growing and continuing to develop. So this is the basic sexual life cycle in humans. At the end of this semester, we're going to talk more about reproduction, and we will get more into the details of this. So let's briefly go over meiosis, which is the production of haploid gametes in male or female reproductive organs. So this happens through 2 complete rounds of cell division. So the phases you will recognize are similar to the phases of mitosis, but we have 2 of them. So for meiosis 1, we have Prophase 1, Metaphase 1, Anaphase 1, and Telophase 1. And then we'll go through a whole second round of cell division, Prophase 2, Metaphase 2, Anaphase 2, and Telophase 2. So meiosis 1 is the first round of cell division, and this is important because this is where genetic variation will be created in the egg and the sperm cells. So DNA replication will happen before Prophase 1, just like in mitosis. So DNA replication happens in interphase. But during Prophase 1, there's a big difference between mitosis and meiosis. So the homologous chromosomes will line up, just like in mitosis, but unlike mitosis, they will actually exchange genetic material during Prophase 1. This is called crossing over, where non-sister chromatids will touch. That's called synapsis. They will break off and exchange genes. So a very odd thing that happens during meiosis 1, which is that the chromosomes touch and they will swap genes. So you will have some maternal genes that end up on paternal homologs, some paternal genes that will end up on maternal homologs. This creates a totally new combination of genes on the chromosomes, and it will mix up maternal and paternal genes on the same chromosome. So here's a picture of that. So we have homologs, maternal and paternal, that have been replicated. They have their sisters. They will actually touch and cross over, mixing up paternal and maternal pieces of the chromosomes, so that these new mixed up chromosomes are now genetically different from the original maternal and paternal homologs. And that's very important because what that does is it mixes up the genes in a single individual so that you pass on a mixture of your genes down to your children. So in other words, this is why you don't just have only your mother's lineage and only your father's lineage to pass on to your children. Your genes will mix up, and then you can pass on combinations of your genes to your children. In metaphase 1, we have another difference between metaphase in meiosis and metaphase in mitosis. In this piece, the chromosomes are lined up and ready to be pulled apart by the mitotic spindle, but in this case, it no longer is specifically lined up so that we get the same maternal and paternal combinations. In other words, they're going to mix up the chromosomes and they will arrange randomly at the metaphase plate, independent of maternal and paternal origin. So in mitosis, you had maternal sisters go this way, paternal sisters go that way. In meiosis, we're going to mix these up. So this is called independent assortment, and it ultimately results in gametes with different combinations of maternal and paternal chromosomes. So here's a view of that. So you will have maternal, paternal homologs lining up during metaphase 1. But instead of all the greens being over here and all the purples being over here, they will mix up and independently assort. So some paternal over here, some paternal over there, some maternal over here, some maternal over there. So it is more variant in terms of which chromosomes or which homologs end up in the daughter cells. So meiosis 1 and meiosis 2 ultimately start with diploid germ cells in reproductive organs. The chromosomes will replicate just like they did in mitosis. But during the process of meiosis 1, the genes will mix up by crossing over. That happens in prophase 1. The chromosomes will also mix up during metaphase 1, such that in the final gametes, you will have a mixture of maternal and paternal homologs that is different than the original diploid germ cell. So why do we care about genetic variability? We care about genetic variability because the paternal and maternal gametes will then combine to give many, many potential gametes and many potential inheritance patterns that can be passed down to the children. So genetic variability after sexual reproduction allows new combinations of genes in offspring that have never before existed. This is because of 1, crossing over, or the mixing of maternal and paternal genes on a chromosome. 2, independent assortment, or the arrangement of chromosomes randomly at the metaphase plate. And then 3, the fertilization from genetically different individuals, a group of genes from mom and a group of genes from dad. So if we do the numbers, humans have 23 pairs of chromosomes, which is 2 to the 23rd possible gametes after meiosis. That's over 8 million possible gametes. 2 individuals will then combine gametes during sexual reproduction, and that can create 2 to the 23rd times 2 to the 23rd, over 70 trillion possible zygotes after fertilization. So this is why you and your siblings are similar, but not identical. And when you do the math, it's actually remarkable that you're similar at all. So don't feel bad if you're very different from your family. Sometimes it'll be trivial, these changes. And sometimes these changes can be the difference between life and death if certain genetic diseases or certain mutations occur during these large changes or genetic variability. So genetic variability over many generations is actually the basis for evolution by natural selection. And we don't have time to get into that, but if you have time to take an evolution and ecology class or a general biology class, I would highly recommend it. This stuff is very interesting. Okay. Good time to take a little break. We are now going to get into DNA, the structure and replication of DNA, and it's going to be a bit. So grab a coffee and grab some notes. Okay, so DNA is the genetic material. So now we understand that the inheritance of traits and the genetic variability occur as a result of chromosome events during meiosis. But what is a gene? How does the actual information transfer happen? When the details of DNA structure were finally worked out, we began to understand the how of inheritance. So here is the structure of DNA. DNA is a long chain-like macromolecule. Go back to our chemistry lecture. Macromolecules are large molecules, and it is made up of subunits called nucleotides. So nucleotides are molecules that contain a sugar. For DNA, that sugar is deoxyribose. A base. For DNA, that base is purine or pyrimidine type. And a phosphate group. So here we have the deoxyribose in blue, the phosphate in yellow, and the purines or pyrimidines in, what color is that? I guess that's pink. The way that DNA is organized is that there are 4 different types of nucleotide bases, which are grouped as purines or pyrimidines. These 4 nucleotide bases are adenine, A, guanine, G, thymine, T, and cytosine, C. The 4 nucleotides act like letters in a code to make up a long string of nucleotides, which will be a gene. And a gene will code for traits like eye color. So if we look over here, this is how one strand or one side of DNA is organized. There's a sugar phosphate backbone, which is made up of sugars and phosphate groups bonded together. So there's your phosphate, and there's your deoxyribose sugar. So phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. This makes a backbone that holds the structure of DNA tight. Off of that backbone, each sugar is attached to a base. A, G, T, or C base possibilities for DNA. So here's a T, that's thymine. Here's an A, adenine. C, cytosine. And G, guanine. All together, the phosphate, the sugar, and the base is a nucleotide, one single nucleotide. But the variable part is the A, G, T, and C, which is why we just shorthand it and write the letters rather than including the sugar phosphate backbone. So the nucleotides are A, G, T, and C. Now I'm not going to have you guys draw out the structure, the chemical structure, or look at the bonding here. Not a chemistry class, although if you take biochemistry, you'll probably look more at this. For now, I just want you guys to know the names and that it's organized with a sugar phosphate backbone attached to the nucleotide. So the phosphate group of one nucleotide is attached to the sugar of the next nucleotide in line. That creates the sugar phosphate backbone. Alternating phosphates and sugars from which the nucleotide bases project. Then one side of DNA will bond to another side of DNA. Again, DNA is a double helix where you have one copy, another copy. They bond together and it twists in a double helix. So the chemical side groups of the nucleotide bases, or the A's, G's, T's, and C's, will bond together from one side of the DNA to the other side of the DNA. That connects one strand of DNA to the other. So adenine forms bonds with thymine, and guanine will form bonds with cytosine. So if you know one strand of DNA, for example, you have an A and a G here, you will always know the other side. It's like a mirror image, and if you have an A, the other side will always be a T. If you have a G, the other side will always be a C, and vice versa. So we can now assemble the two sides or the two strands of DNA, where you have a sugar phosphate backbone on one side with the nucleotides projecting out. Here's A, C, G, and T. And knowing that strand, we can then put a mirror image strand based on which pairs go together. So you have a sugar phosphate backbone now on the other side, but it'll be flipped in a mirror image. And the bases projecting from that will be determined by the bases on the first strand. So if you see an A, the next base will be a T. If you see a C, the other base will be a G, etc. G, then it's a C, T, then it's an A. Get it? So these strands are mirror images of each other, and one is flipped with respect to the other. At the very end of a sugar phosphate backbone, we always have a hydroxyl group. We call the phosphate end the 5', and we call the hydroxyl end the 3'. So we have a phosphate backbone from a 5' to a 3' end, and then we have the bases. The other side of the DNA strand will be flipped, and it's going to run from phosphate at the bottom to hydroxyl at the top. So it runs from 3' to 5', and its bases are determined by the bases on the first strand, which will be complementary to the others. We say this is anti-parallel because you have 5' to 3', and then 3' to 5', bonded together by their bases. So here's a review of DNA structure, but I think it would also help for us to draw it out. So let's draw it out. Okay, so DNA structure is like this. You have phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, to make the backbone of DNA. Now the first phosphate is at the 5' end. The last phosphate sugar group is capped off by a hydroxyl or an OH, and we call that the 3' end. So we have the sugar phosphate backbone, and off of the sugars we have the bases. And the bases in DNA are A, G, T, and C. We'll have a long string of bases which will encode a particular trait or a gene, for example, eye color. This is one strand of DNA, and it will be bonded to a mirror image strand that runs in the opposite direction. Whenever you see an A, you add a T. Whenever you see a G, you bond to a C, and vice versa. When you see a T, you bond to an A. Once you see a C, you bond to a G. When you see an A, again, you bond to a T. Okay. And then we know there's also a sugar phosphate backbone on the other side. Okay. The difference here is that this sugar phosphate backbone runs in the antiparallel or opposite direction, such that this phosphate down here is the 5' phosphate, and then up here we cap it off with a 3' hydroxyl. So we have one strand of DNA that runs 5' to 3' with its string of nucleotides, and we have a complementary strand that complements the bases with the base pairing rule that runs in the 3' to 5' direction. And we'll read DNA like this for each of the 2 strands. So again, we have the 5' to 3' and the 3' to 5' strands. We wind those together to make this double helix of DNA. So the complementary base pair rule, which I have already talked about, but I want to just formalize it here, is that A always bonds to T and G always bonds to C. This is what gave Watson and Crick the Nobel Prize. If you've heard of Watson and Crick, they discovered this complementary base pair rule to finalize the structure of DNA. Lots of history there and very interesting. In other words, what they figured out is that if we have one strand, here I just randomly made up a strand, AGTT, CCTT, okay, long strand, which will give us a gene, you can use the base pairing rule to know that the second strand will be the exact complement of that. So everywhere you see an A, you put in a T, G, C. That's nice because now we only need to figure out one strand, the sequence of one strand, to know the sequence of the entire region of DNA. We call these coding and non-coding strands. So as it turns out, one strand of DNA is the code that is used later to actually make the proteins. This is the sense or coding strand, and it's the 5' to 3' strand. Randomly, there's the sequence I made up for you guys. The other strand is a nonsense code. That is that antiparallel or mirror image that's used to stabilize the structure of DNA, so they bond together and stabilize in this double helix. It also helps with replication. So we'll copy one side, then we'll copy the other side. And that nonsense code is the 3' to 5' strand, or the non-coding strand. And there's the mirror image template strand, mirror image non-coding strand, which matches exactly with the template. Okay, now we know enough to talk about DNA replication. So for DNA replication, this is going to happen when? Do you guys remember? What phase of the cell cycle? Interphase. So it happens at the end of interphase, just before we're going to divide the nucleus in mitosis. So it happens in interphase, and then we go through the phases of mitosis: prophase, metaphase, anaphase, telophase, and then cytokinesis, right? Okay. So this is all happening in interphase before mitosis, or before the cell division. And what we do is we take a single strand of DNA, use that as a template, add new nucleotides to that template to copy one strand. Then we will have the other strand, and we'll add nucleotides to the other strand, copy the second strand. We end up having 2 identical copies, one of one strand, one of the other, which gives us 2 new DNA helices. And I'm going to separate them in 6 stages for DNA replication. Okay, so I'll list them here, and we're going to go through each one and talk about the enzymes involved and the specific processes that happen during DNA replication. Okay, so step 1 is to unwind the double helix. We have this double helix of DNA. We need to put in an enzyme to open up those 2 strands. So we have the coding strand and the non-coding strand. In this diagram, you'll see, although it's twisted, so it's complicated, we have the 5' strand here to 3'. So the coding strand is the 5' to 3' strand. The non-coding strand is the 3' to 5' strand. We're going to add this enzyme, which is called helicase, and it's going to open up and unzip the DNA double helix. So there's this really nerdy joke for DNA replication, and that is, what do teenagers and helicase have in common? And the answer is they both like to unzip things. Yeah, I know, we're just trying to keep you guys awake. Okay, so helicase comes in and it unzips or unwinds the double helix. So now we have separated the coding and non-coding strands, the 5' to 3' strand, and the 3' to 5' strand. We call this the replication fork. The second step is to now prime or begin the process of adding nucleotides. We start on the 5' to 3' strand and we call that the leading strand. The enzyme that adds the primers is called primase. So the first few nucleotides are added, and a picky little detail here is that actually these primer nucleotides are RNA nucleotides, not DNA. And we're going to add these primers to the coding strand, the 5' to 3' strand first. So one hint for this diagram is to read it from right to left. You notice that the arrow here is moving from right to left, so the primer is added first at the 3' end. As the primers are added, they are complementary nucleotides. So if you see an A, you will add a T, or actually because this is RNA, you'll add a U. We'll get into that later. If you see a T, you will add an A, and so on for G's and C's. So step 2 is priming. We're going to add a few RNA nucleotides to start the process of adding nucleotides to the coding strand. The next step is to start adding DNA nucleotides. DNA nucleotides will be added just to the first coding strand or the leading strand of DNA. This happens quickly, starting at the 3' hydroxyl group and adding the sugar phosphate backbone and the complementary bases. And we'll continue to add the leading strand with complementary bases with this enzyme, DNA polymerase 3. So DNA polymerase 3 is the enzyme that adds the DNA nucleotides. So we've got primase, which is going to add RNA nucleotides that starts the process, and then DNA polymerase 3, which is going to add the DNA nucleotides. We'll get to this one in just a second. The helicase continues to unzip or open up the DNA double helix as the nucleotides are added to continue the process through the whole strand. So, as I said, this happens in the anti-parallel fashion. So we start with the 5' to 3' strand, and we're going to add complementary nucleotides with DNA polymerase 3. So notice that here is our 5' to 3'. I know it's kind of flipped sideways. We're going to add, if we see a C, DNA polymerase 3 is going to add a G. If it sees a T, it'll add an A. A, it'll add a T. C, it'll add a G, etc., including the phosphate backbone. So you get an entirely new strand that is complementary to the template strand. Step 4 is that once the leading strand is on its way, we will then go to the lagging strand and start adding nucleotides to the lagging strand. We call this the lagging strand because it's slower and it doesn't go as smoothly. So this is going to be the non-coding strand that runs in the 3' to 5' direction. And it's going to be added in the same fashion, but the enzymes don't like to work in this left to right direction. So it adds a chunk and then another chunk. It starts with a primase where it adds the RNA nucleotides to start the process, and then DNA polymerase where we add the DNA nucleotides. So we're adding nucleotide, 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 but then it pauses, and then we start over again. Because of these pauses and because it's moving in this left to right direction, this is a slower process that is a lagging strand. It happens in smaller chunks as opposed to the continuous fashion that occurred on the leading strand. This is also done by DNA polymerase III to add the nucleotides. In the final step, we had the leading strand and the lagging strand with their nucleotides complete. We now have to remove the primer or RNA segments. DNA polymerase I is the enzyme that does this. So it's going to remove these primers and replace them with regular DNA nucleotides. Notice that DNA polymerase III is used before DNA polymerase I, so they're not numbered for the order in which they occur in the process. They're numbered for, I'm guessing, the order in which they were discovered. So I didn't name them, don't blame me. DNA polymerase III happens first, and then DNA polymerase I comes in and replaces all of the primers with regular nucleotides. Finally, we need to join and rewind the new copied segments of the, I'm getting tired, the new copied segments of the DNA strands. So here, let's look at our leading strand. We copied and formed a new anti-parallel strand to that leading strand. We now need to join and rewind this new double helix. DNA ligase is the enzyme that does this. So what enzymes did we have? We had helicase that opened up the DNA, primase, which added the RNA primers to start the process, DNA polymerase III, which added the DNA nucleotides, DNA polymerase I, which replaced the primers with DNA nucleotides, and then DNA ligase, which joins and rewinds the helices back together. When we do that, the process of DNA replication is completed, and we have two new identical copies of the homolog chromosome. So this would be one sister, this would be another sister chromatid that is now replicated, so we can start the process of mitosis in prophase 1. So as you may have noticed, this is a complex process, and DNA is a very, very large molecule. So it's actually far too big to have just one replication fork. So replication actually will occur in segments of about 100,000 nucleotide chunks at a time. With multiple replication forks and multiple leading and lagging strands, adding, joining, rejoining, it's actually remarkable that errors are very rare in this process. It's very important that the DNA is faithfully copied from one cell to the next. Each cell of replication, though, introduces a new possibility of errors and change in the DNA sequences. That's why if you go back to mitosis, we have these checkpoints to make sure that it is properly replicated. Any change in the DNA code is a mutation. Now, mutations can be caused by replication errors. They can also be caused by a couple of other things. They can be caused by chemicals, for example, cigarette smoke, radiation, and UV light. They can also be caused on purpose during the process of meiosis. During genetic recombination or crossover, we can get new changes in the DNA, which would technically be mutations. There are a lot of types of mutations. Change in one or just a few change in one nucleotide is called a point mutation. So for example, if you started out with ATGC and you had a quick mutation where the G turned into a T, that's called a base substitution. You get ATTC. You can also have a deletion. So if you went from ATGC to delete the G, you have ATC. You can have an insertion where you insert a new base, ATGCC. This can cause minor changes, usually base substitutions are minor changes, or major changes in completely shifting the gene in the case of deletion and insertion. So change in a large segment of DNA is gene transposition. An entire gene can move to a totally new place on a chromosome or onto another chromosome, and that's called chromosomal rearrangement. So a single change in a nucleotide can sometimes cause an entirely different variant of a gene, green eyes to blue eyes kind of thing. It can sometimes completely alter a gene's function, or it may even render a gene unreadable. So blue eyes to green eyes, not a big deal, but sometimes it can mean genetic disease or more serious mutations. So the possibility of change from one error is a very wide range, and sometimes mutations can be serious. They can be deadly in the case of genetic disease. They sometimes can be helpful. They can lead to new traits that are advantageous. So let's say, the ability to produce melanin, which can protect your skin against sun damage. That would be advantageous. Sometimes they have no effect at all, and you won't see any changes in the individual. So a lot of things can come from DNA mutation. Here's some examples of single base substitution mutation. So mutations can occur in any cell type, anywhere where you have DNA replication, so during the process of mitosis, in somatic cells can cause mutation. These can have really drastic effects for the individual, for example, lung cancer as a result of cigarette smoke, but they won't be passed on to that individual's children. They won't be inherited because they're in the body cells. So it can lead to cancer and other problems, but it won't be inherited. Mutations in reproductive cells, however, can be passed on to offspring and can be more serious. Okay, I think that's enough for part 1 of DNA. Take a little break, come back. We're going to do part 2 of DNA and talk about how DNA and the sequence of nucleotides actually becomes a final protein.